Hello, today we're going to be talking about microscopes. Um, this is just a little bit of history about the microscopes. Anton van Leeuwenhoek was kind of the father of microbiology. He was really the first one who used the microscope, and again, very simple microscope. But he was the first one to look at the single-celled organisms. Okay, so this simple microscope was used in the 1600s, and it was very, very simplistic. Technology has really increased, and that has made such advances in science. So as we go a little further, after we've used a little bit of more technology, Robert Hooke looks at cork cells, and he says, this looks like the cells of a monastery. So the monks would live in these little rectangular rooms, and that's how he termed the coin cell. And we'll talk a little bit more about that as we go through the cell. So a microscope is an instrument consisting of a lens or a combination of lenses, and that's what we have, a combination of lenses, that's going to produce an enlarged image of a small object. We have two types of microscopes that we will use in class. We have a compound light microscope and we have a stereo microscope, or it's called a dissecting scope. We will also learn about two additional microscopes. We have a transmission electron microscope and a scanning electron microscope. Okay. When we talk about microscopes, we need to define the term resolution. Resolution is the ability to distinguish between two objects that are next to each other. Where I grew up, it was a town called Manville. The town was separated into the north and south side by train tracks. So I lived on the south side. I needed to cross the train tracks every day to get to school. And as a kid, we spent a lot of time walking along the train tracks. And one thing I do remember is we would look down the path of the railroad tracks. And at a certain point, I couldn't tell that there were two rails of the train tracks because they merged together. So at that point, I did not have very good resolution. So I did not have the ability to distinguish that there were two different train track rails. Okay, so we're going to be seeing that as we do the microscope lab. The microscope that we use in class is called the compound light microscope. Okay, so this is going to use visible light, hence the light component of the name, to illuminate the object being viewed. So we're actually going to be plugging in the microscope and a light will turn on so we can see the object we're viewing. Okay, it's called compound because it has a double set of lenses. It has a lens at the eyepiece and then there's a lens in what we call the objectives. Okay, now this is pretty impressive. It will allow magnifications up to 1,000 times. There is a little slight problem. It's not that great for research labs and high-tech needs because it cannot resolve objects that are closer than 0.0002 millimeters apart. Okay, so it can't tell that there are two objects close to each other at that distance. For us, it's no problem. And that's what it would look like. We will be able to see all of this in class, and I will define all the parts, and you will be actually learning about the microscope through lab. This is what a fruit fly would look like underneath the microscope. This would be a euglena. A euglena is a type of a protist, and we will actually see this live. Okay. The stereoscope is often called the dissecting scope. It's very similar to the compound light microscope because there's light. You're going to plug it in. We do have a couple of lenses, but it's much lower in magnification. One benefit for this is that the distance, it's called the working distance from the specimen to the lens, is bigger. So you could put bigger objects on it. And that's what that would look like. And again, we'll learn about this in class. The fruit fly, that's what that would look like. Okay, this is the surface of a leaf. Okay, now we do have two types of electron microscopes. We have a transmission electron microscope and a scanning electron microscope. Okay, now compound light microscopes use light to view the objects. For electron microscopes, it uses electrons. So for a transmission electron microscope, we need to freeze the object, slice it into very, very thin sections, put a beam of electrons through the object. Now, Electrons need to be focused in with magnets, and then it gets projected onto a screen. Okay, so as far as the procedure, that is what, I'm sorry, not the parts, excuse me, that is what it would look like. The object has to be frozen, so nothing can be living. All right, it has solved the problem of resolving small objects, and it was invented in the 1940s. It can resolve two objects that are as close together as 0.0000005 millimeters apart. Okay, and the magnification, just to give you how amazing it is, it would stretch a sneaker from Boston to New York City. Okay, this is another type of a protist, okay, with a different magnification. This is the euglena, and if you're noticing on these pictures, we are focusing on internal structures. Scanning electron microscope shows a lot of detail. It's going to take a picture of the surface of the object rather than taking the thin slices and having a beam of electrons pass through it. So with the scanning electron microscope, we take a picture of the objects, the electrons sweep over it, magnets are used to focus it, and it is projected, but it will give a 3D quality. The magnification is a little less than the transmission electron microscope. The machine is quite big, but again, you're showing a different part of the organism. So that is that protist that we saw earlier. There's that euglena, and again, as you're seeing the pictures, we're focusing in on the surface. Okay, and this chart just gives you an idea of size of things. So if you have any questions, please see me tomorrow. Thanks.